I just need you to look at this. This might be one of the most incredible structures in all of biology. We're flying over a near crystalline layer of proteins protecting one of the most extreme organisms on the planet. This is Deinococcus Radiodurans, a hardcore bacteria that's been in the news a lot lately for being basically indestructible. For real, since its discovery back in the 50s, scientists have thrown basically everything we can at samples of this little fella. It's been blasted with thousands of times the lethal dose of ionizing radiation, and survived. They had a sample of this stuff live on the outside of the International Space Station for a full year, totally fine. And most recently, there's this paper making the rounds where an especially eager team from Johns Hopkins University exposed D. radiodurans to enough force to simulate an asteroid impact, which this bacteria effectively shrugged off. I mean, it's kinda hard not to call this thing a bacterial spaceship. But the closer we look at this astounding little critter, the more we find Earth-based forces that would push this kind of system to evolve. So today, let's explore the incredible biology that makes Deinococcus radiodurans one of the most durable living things on the planet. Let's get into it. And hey, welcome back to Clockwork, a show about the incredible details at the molecular foundations of life. Make sure you subscribe so you don't miss out on new episodes, and head on over to patreon.com/clockworkshow to check out our awesome sticker packs and molecular papercraft scenes. But for now, let's pick apart this tank of a bacteria. So yeah, Deinococcus radiodurans is an incredibly well-studied extremophile bacteria. It was first discovered in the 50s when scientists in Oregon tried to sterilize a sample of dog food with a huge dose of gamma radiation, which D. radiodurans survived. Since then, this fella has baffled the scientific community with the ability to withstand thousands of times the radiation dose that would instantly kill a person. Hence the name, durable ANS, right? Cool. And here's the thing, radiation resistance was the focus of research into D. radiodurans for ages, so a lot of what we're going to see in this video is focused on surviving Hulk-level doses of gamma rays. But D. radiodurans is a scavenger. It lives by landing on a pile of trash, pumping out a bunch of digestive enzymes, and slurping up the nutrients that come back from that. Surely this bacteria didn't evolve the ability to survive meteor impacts and the vacuum of space just to find more trash to eat, right? But we're going to be able to put together a much cooler picture of why radiodurans ended up this way as we examine each individual mechanism. In this video, we'll pick apart a handful of the mechanisms Deinococcus uses to stay alive no matter what the universe throws at it. But anyway, let's actually start exploring what makes this bacteria so tough. This is roughly what the average Deinococcus radiodurans cluster will look like. We've got 4 of these little guys kinda huddled together in this hairy little blob, and usually you'll find them on a pile of dead organic material where they scavenge for food by pumping out digestive enzymes and absorbing what carbs and protein they can from their environment. Like I said before, D. radiodurans is immediately striking because instead of just an outer membrane like other cells, they cover themselves in this near crystalline protein armor. This is the S layer of Deinococcus radiodurans, and it is mainly composed of one protein, HPI. HPI is made out of 6 little chains like this, which, by the way, are made out of egg-like domains just like the main structural component of the titin protein in your muscle. A full HPI unit is pumped out of the D. radiodurans membrane, and 6 subunits of this HPI armor will self-assemble into this sturdy-looking snowflake complex. And a bunch of those complexes will link up into this sheet that covers the entire bacteria. And look at that! They're little hexagons. Wild! I wonder if that shape has any, special mathematical significance or anything. Anyway, this layer of hexagonally packed intermediate protein also serves as an anchor for long chains of sugars called Deinopol. These form a kind of cell wall for D. radiodurans. Now, the exact structure of Deinopol has yet to be determined, but researchers have proven that these sugars provide some defense against radiation as well as an antioxidant buff. Even at their edges, D. radiodurans is going for maximum radiation shielding. While this hexagon shell looks incredible, it's also incredibly expensive to make. What's more, this shell doesn't seem to directly contribute to radiation defense. Instead, the S layer allows D. radiodurans to hold its shape no matter what hits it, even a meteor impact, and it also seems to help defend against big temperature swings. Sure, it serves as an anchor for this radioprotective sugar shield, but you can find way cheaper methods to anchor a cell wall to your membrane as a bacteria. But yeah, that's a really interesting distinction that may help us understand what D. radiodurans is actually trying to survive later on. Keep that information in your back pocket, I guess. For now, we can find some direct radiation defense just below the S layer, where we find these beefy pore complexes. Just beneath the S layer are these SDBC complexes embedded in the outer membrane. These are where the radiation defense gets kicked up a whole new level. Right off the bat, these 3 pores are studded with a powerful antioxidant called dinoxanthin, which adds its own form of strong radiation defense. And just a quick fun fact, dinoxanthin also contributes to making D radiodurans a light pink color, which is just neat. But moving on, just beneath these pores are a trimer of an even stronger antioxidant domain, which is a superoxide dismutase, which provides an even stronger radiation buff. Now, you might be wondering why I keep describing these S-layer proteins as antioxidants when I'm supposedly talking about radiation defense. And trust me, the connection between those two ideas is going to blow your mind if you don't already know it. But stick with me here. We'll get to that connection once we have a few more pieces of the radiodurans puzzle. Going deeper, the SDBC complex actually has this cool tail that extends all the way down through D radiodurans' outer shell. This bacteria uses a classic double membrane, with just a ton of peptidoglycan in between to hold its shape. With SDBC tethering the inner membrane to the outer membrane, that structural support is even stronger. All in all, the S-layer and anti-radiation carbohydrate coat of D radiodurans provide a great defense and a passive buff to radiation damage. But in order to survive the level of gamma rays that Deinococcus radiodurans does, you're going to need a lot of active countermeasures as well, which we're going to get to right after this. I've loved making this episode so far just because of how cool Deinococcus radiodurans looks. While this S-layer looks incredible in animations, it looks even better in paper. That's right, we'll get back to the video after a word from this video's sponsor, which is, once again, me. Sorry. This video is brought to you by our incredible supporters at patreon.com/clockworkshow. Joining our tiers gets you access to tons of cool physical perks like a monthly sticker pack subscription and our growing Papercraft Club. This month, our Papercraft members are receiving either their own 8" by 10" section of a molecularly accurate Deinococcus radiodurans S-layer, or our Minicraft subscribers will get a single higher resolution hexagonal subunit from that same S-layer. We're coming out of a difficult period here at Clockwork HQ, and this project has only been able to survive a few setbacks because of the incredible support we get from our patrons. Visit patreon.com/clockworkshow and subscribe today. And now we can get to the good stuff. So we've seen the great perimeter defenses employed by D radiodurans, but now let's go to the bacterial interior and see where the real magic happens. If there's still radiation getting through this S-layer, how does Deinococcus radiodurans mitigate that damage? Cutting directly into one of these little fellas gives part of the game away instantly. Deinococcus radiodurans is optimized for durability. While other bacteria make a soupy mess of their DNA, D radiodurans organizes its DNA into this torus. Donut shape. While this nucleoid looks cool, and it's fun to say donut DNA, the circle structure isn't the most important aspect here. It's what radiodurans packages inside this donut. D radiodurans maintains multiple copies of its genome and several redundant copies of critical genes. More importantly, those copies are persistently linked by structures called holiday junctions, which are involved in a repair process called homologous recombination, which means that even when this bacteria gets blasted with a ton of DNA-cutting radiation, it can cross-reference one homologous chromosome with another and fix DNA breaks with remarkably few errors. D radiodurans is basically always on the lookout for DNA damage and is set up to be world-class at DNA repair. But let's explore what that means. Now, we could either spend the next 30 minutes going over the mechanics of D radiodurans' vast DNA repair arsenal, or we could quickly cover the highlights and I can one day produce a very labor-intensive Part 2 video doing a much deeper dive. Hit me up in the comments if you really want to know the nitty-gritty about how Deinococcus radiodurans' DNA repair goes down. For now, recombining DNA is radiodurans' main weapon here. Radiation, and other things, primarily kill you by causing double-stranded DNA breaks, that is, cutting through both helices of your DNA molecule. Deinococcus radiodurans has a robust damage detection system, and the star of the show here is a protein called Rec A, which forms these little fibers around damaged DNA, and then helps find a matching sequence inside the duplicated chromosome, which other enzymes can then use to synthesize a repaired DNA strand. Now, the details here are a little hazy, but Rec A sometimes can team up with another protein called DDRB and its partner DDRA to help single-strand DNA breaks in a process called synthesis-dependent single-strand annealing. And a lot of scientists think this is the main superpower D radiodurans has for efficiently repairing DNA breaks. Meanwhile, scientists studying populations of D radiodurans that lived on the exterior hull of the International Space Station for a year found that surviving bacteria upregulated these UVR proteins. The UV radiation in the hard vacuum of space forms all sorts of goofy-looking lesions on DNA like this, and the UVR family of proteins does something called nucleotide excision repair to clean up the breaks. I really could keep going. The map of DNA repair in Deinococcus radiodurans alone is just wild and incredible, but I think it's best to just put all these systems in context. The upshot here is that Deinococcus Radiodurans can survive and repair hundreds to thousands of double-stranded DNA breaks, while most other cells only have the capacity to fix a few dozen. And this robust DNA repair suite only works because of all these DNA redundancies our hero packages into its nucleoid donut. Essentially, it is really unlikely that the exact same DNA break is going to happen across every copy of D. radiodurans' genome. Even if there are multiple breaks across every copy, Radiodurans will just recombine all the damage and end up completely healed. This means that D. radiodurans not only can survive radiation exposure, but actually thrive under it, still growing and dividing even when it's actively being irradiated. A fatal dose of radiation for you barely counts as background noise for this fella based on the way that it's optimized its living strategy. If you're asking how this is possible, one review paper I read chalked this up to an investment strategy. Both the S layer and this robust DNA repair system cost a ton of energy. Most other bacteria invest energy into growing and dividing as quickly as possible, while Deinococcus radiodurans is making more of an efficiency play. D. radiodurans grows and divides much slower than regular bacteria, and they're also a little bit larger too, but it also has a much higher chance of actually passing its genes on because it can survive so much more. It's like a quality over quantity play, basically. And that's all fun, but let's take this to the next level, because I've left a few details out here. Here's the big question. Do you know how radiation causes DNA damage, and therefore, damage to you specifically? I've talked about this mechanism a little bit in previous videos. In short, while the high-energy rays of ionizing radiation can cook DNA on their own, direct damage from ionizing radiation only accounts for about 20% of the damage it does to our DNA. The rest, 80%, is indirect, because ionizing radiation will bump into just about anything, especially the water that these cells are made out of. Hit a water molecule in just the wrong way and you can break it, creating truly violent forms of oxygen called reactive oxygen species, or ROS. These rabid oxygen variants will flail wildly and do huge amounts of damage to just about anything they bump into, which is why Deinococcus radiodurans uses a deep antioxidant pool. That's right, the antioxidants in the D. radiodurans S layer are there to absorb the harmful ROS byproducts caused by radiation, and other things. I'll get to that later. Two of the worst ROS characters are the superoxide ion and hydrogen peroxide. D. radiodurans employs a bunch of different superoxide dismutase enzymes that disarm ROS species before they can latch onto and damage something else. This is why antioxidants are such a big deal. Our metabolism naturally produces a ton of reactive oxygen species as a byproduct of, using oxygen to breathe. The wild thing is, radiation isn't the only thing that kills you with reactive oxygen species. Heavy metal poisoning and other toxic chemicals can also cause cytotoxicity by initiating a ROS cascade. In a lot of ways, Deinococcus radiodurans isn't trying to defend itself against radiation, it's trying to defend itself against reactive oxygen species. And that's going to help us unlock the final piece of the puzzle here. It's cool that radiodurans here can repair its DNA and soak up a ton of ROS damage, but what's stopping all these harsh conditions from breaking the repair machinery too? And as an aside, this is one of the ways that research exploring Deinococcus radiodurans has unlocked our understanding of radiation damage itself. Because it's not the DNA damage itself that kills cells when irradiated, it more and more looks like it's damage to cell proteomes. Turns out, radiation and reactive oxygen species also cause a ton of damage to the protein workers in cells as well. Protein damage means that the DNA repair system will break down, which also means that busted enzymes won't get replaced if they break too. So we're stuck with a big question. How does D. radiodurans shield its proteins from getting damaged in the first place? Antioxidant proteins can only do their job if they're in the right shape. How can this bacteria thrive under consistent radiation exposure? And the bacteria employs one more passive buffer to tip the scales in its favor. And that buffer is simple. It just maintains a high concentration of manganese ions. That's right, folks, meet manganese, my personal favorite metal on the periodic table. Manganese ions power a lot of our biology. In fact, some researchers figure that the earliest life on our planet ingested natural manganese clusters as a kind of primitive radiation defense. For me, most importantly, manganese forms the core of the oxygen evolving complex in photosystem 2, which is the molecular ground floor our entire biosphere is built on top of. And based on this poll, manganese basically gets no respect from my audience. And this bit would be far more effective if I personally didn't misspell manganese in the post where I was trying to prove this point.
Manganese.
So like even manganese's biggest supporters don't give it enough respect. Anyway, the oxidation state of easy-to-find manganese ions makes them great at absorbing the electrons on things like the superoxide reactive oxygen species. So having a lot of manganese inside your cell and also maintaining a high manganese to iron ratio makes for an incredible first line of defense against radiation damage that helps guarantee that your critical DNA damage repair enzymes don't get kneecapped when oxidative stress arrives. If you want to learn more about manganese, check out the episode MQ Tate did on the element. Heck, check out his entire ongoing periodic table series. When it comes to analytic chemistry, MQ Tate is the best and I will accept no substitutes. But yeah, D-radiodurans maintains up to hundreds of times the manganese concentration you would find in other cells. But manganese alone won't cut it. No one is suggesting that you could just down a manganese supplement and then walk into the reactor at Chernobyl or whatever. Instead, our hero also makes these short little peptides that spontaneously form passive antioxidant complexes by accumulating manganese ions, phosphate, and other metabolites into these ephemeral little concentrations. During times of radiation stress, these pockets accumulate around D-radiodurans' DNA and DNA repair proteins, forming an incredibly powerful passive buff against ROS damage. Meanwhile, D-radiodurans takes this manganese buff to the next level by pumping manganese ions out of its membrane and into the S layer and sugar shield, supercharging this region's ability to soak up radiation and reactive oxygen species before that stuff can even penetrate the cell. And that kind of puts the whole thing together. Deinococcus Radiodurans can survive extreme radiation doses thanks to the interplay of a bunch of tiny factors that add up to an incredible balance. Deinococcus Radiodurans is out here defending every protein it can from damage so it has the best chance to keep going. It's not just one thing that makes Deinococcus Radiodurans so powerful, it's the interplay between all these systems. Researchers describe Deinococcus Radiodurans as a remarkable generalist. This bacteria just operates in a way where it can take a bunch of huge hits and keep going, which has allowed it to survive basically anything that life on this planet can throw at it. But, you know, we have all those details, but that hasn't really answered the questions I've set up here. Why does D-radiodurans need to survive thousands of times the radiation dose that would insta-kill a person? I mean, Earth has been radioactive in the past, and the sun's a deadly laser and all, but nothing has been this radioactive to necessitate this much of a defense. What are the actual evolutionary pressures that would make something like this evolve? And remember, radiodurans here isn't trying to survive radiation, it's trying to survive the damage caused by reactive oxygen species. Don't forget, a lot of these proteins up in the S layer are also helping our hero maintain its shape. Radiation hurts, but it definitely doesn't hurt like a hammer. And the confusion here comes mostly from how we framed our exploration of Deinococcus Radiodurans. We're thinking in terms of radiation defense because that's a kind of cellular damage that's relatively easy to throw at model organisms, and therefore that was the question that a lot of researchers were asking back in the day. So let's expand our focus a little and try to complete this puzzle. Here's one final piece that can help us tie this all the way together. Another protein class that's key to radiodurans' survival are these LEA fellas. When things get hectic, these little LEA proteins will condense into these long helices and help cells maintain their shape. But what's the stress that LEA proteins mitigate? Check this out, it's desiccation. You know, drying all the way out. Mummification, whatever you want to call it. And now we can start really putting this together. Turns out, in the 90s, a team of researchers tested a bunch of the radiation defense proteins employed by D-radiodurans and found that those factors also significantly protected against desiccation, which is incredible. How does that even work? The theory here is that as cells dry out, these proteins will condense into the long helical chains you see here and prevent other proteins from clumping together and stabilize membranes once the internal water pressure gives out. Once again, maintaining structure is another really important piece of D-radiodurans' survival puzzle. And this is enough for me to start pulling together a more plausible picture of what exactly D-radiodurans developed all these systems for. Here's the thing, losing all your water does a lot of crazy things, and one of them is produce a ton of reactive oxygen species and DNA damage. Which means we've been looking at this problem all wrong. Radiation defense is just a nifty side bonus of D-radiodurans' astounding ability to continue living and thriving while the world around it completely dries out. Which brings us to a model for what exactly drove Deinococcus Radiodurans to evolve all these interconnected systems. At its core, Deinococcus Radiodurans is a scavenger. It's going to lazily soak up food from dead things and not cause too much drama. Other organisms are going to spend a ton of energy and compete for food and resources, which makes things a little more difficult for our hero. But if the environment completely dries out, then everyone else dies while D. Radiodurans can use its repair systems and interconnected antioxidant shields to legitimately thrive even when all its rivals have effectively turned to dust. When water returns, Radiodurans will still have a relatively empty playing field to thrive in before any competition has time to get established. And I love this view. I mean, sure, it's cool that D. Radiodurans can, entirely by accident, survive astounding levels of ionizing radiation, but I love so much that this bacteria developed this incredible tapestry of interconnected coping mechanisms simply because this guy refused to die of thirst. And you know what? I guess the way I framed the title of this video might be a little too clickbaity now. We've got an entirely Earth-based explanation for why D. Radiodurans lives this way. I guess I'll just have to sit back and take all the angry comments now. Except, there are a few annoying details that don't line up perfectly with D. Radiodurans just surviving desiccation. There's this overemphasis on UV protection that goes beyond just the damage you would get from drying out in the sun. And why does Radiodurans need to make a cell wall out of like radioprotective cotton candy if we're worried about the Ross generation inside the cell? While research into this bacteria is still ongoing and we're still actively investigating potential answers here, very late into the process of making this video, I stumbled into this review paper put out by the Cold Spring Harbor Laboratory that briefly laid out a theory that ties everything together for me. D. Radiodurans is ubiquitous worldwide. And there are a lot of ways that can happen, but here's my favorite idea. Some bacteria form spores and get swept up in air currents, blown as high as the stratosphere, and then are brought back to Earth by rainstorms. Now, while Deinococcus Radiodurans doesn't form spores, it has shown that it can thrive while desiccated, which would make it even easier for air currents to carry it to altitudes where UV radiation becomes more and more problematic. Up in the stratosphere, the bacteria would be exposed to up to 1,000 times more potent levels of deadly UV radiation from the sun. And since rain is the primary vector for bringing airborne cells back to Earth, the return journey will very likely lead to a place where there's more food sources for D. Radiodurans to decompose. It all fits together. So, in the end, all this defense might be a brilliant migration strategy on the part of Deinococcus Radiodurans. The easiest way to find new food sources is to get taken there on air currents. And this finally helps us understand the S layer a lot better, because again, the main thing it provides is a way to survive temperature swings and structural support, which are two things that become a huge problem once you're fully desiccated and also getting yeeted into the upper atmosphere. To summarize, Deinococcus Radiodurans evolved to survive drying all the way out and then getting blown to a new food source, and that just had the side bonus of also defending against the mechanism that radiation damage uses to destroy our cells, which I think is just incredible. While there's still so much to learn about how and why Deinococcus Radiodurans lives the way it is, the hilarious part of me making this video is discovering that yes, it is entirely plausible that Deinococcus Radiodurans evolved effectively the equivalent of space travel, just to have a better chance of finding new and interesting patches of trash here on Earth. Now, while even the highest parts of the stratosphere aren't even halfway to the technical definition of space, maybe you could indulge me here real quick, Now, there's just so much more to be discovered about Deinococcus Radiodurans, but this is a decent enough introduction. I am never not amazed by all the different niches life can carve out for itself on this good Earth. Our planet is astonishing in the circumstances and chemistry it can produce, and for me, it is more than enough to account for all the wondrous forms life can take. Even though a lot of the research around Deinococcus Radiodurans tries to cast this as a space bacteria or a Martian bacteria that landed here, all of these proteins and all of these systems are found across our tree of life, and all of Deinococcus Radiodurans' systems can be explained by evolutionary pressures here on this planet alone. Life on this Earth is astounding in its complexity, and you're always going to find new and interesting pockets where, if you look deep enough, you will be truly blown away by the incredible ingenuity of incremental change through evolution. For me, this has just been a really fun way to explore some really big subjects here in this revolutionary moment in molecular biology. But that's a good place to end it, though. I really appreciate your time. Thanks so much for being here. And hey, thanks so much for making it all the way to the end of the video. If you like this video, please make sure you like and subscribe. I can't express enough how much that helps. At the same time, make sure you check out all the cool stuff we have over at patreon.com/clockworkshow. That's the best way you can guarantee that we're able to keep producing these videos right now. Otherwise, head on over to watchclockwork.com to sign up for our free newsletter. Either way, the very best way you can support this project is being right here, right now. Watching a video all the way through is such an incredible help. Thank you so much. For now, here's a weird one. Do you have a favorite kind of mushroom? If you do, let me know in the comments. I'm trying to see something. I was overjoyed when most everyone gave me the objectively correct answer to the question I asked in our last video, and I want to see where this one takes us. But either way, thanks so much for being here. I genuinely appreciate your time. As always, I like to leave you with peace, love, and a high manganese to iron ratio. Everyone be well. Thank you so much.
Manganese.